If, as we attempt to envisage and vision space media illustrates, there are two independent raw data potentials, an incoming field potential together with an explicit detail potential, being collected at the retina where conventional theory considers there to be just one, we can assume that much of what is currently being attributed in terms of receptor function and retinal circuitry would be in need of fundamental review. We can only make an attempt at this in outline here, but the process should throw up directions for research to either confirm or deny. Our top-down assumption suggests that decoherence is taking place at receptor level, and that both resulting phase and particle potentials are segmented for independent propagation and further analysis. These data potentials break out in terms of perception pathways broadly as follows. Firstly, an holistic and implicit form of spatial awareness generating proximity cues with motion comprehended in flow, then a form, colour and detailed explicit take on reality with space through depth perception and motion comprehension via tracking. This would entail the retinal membrane segmenting both data potentials from the incoming light array via receptor specialism, and then supporting the subsequent circuitry for their development to a point where they are independently streamed to individual areas of the brain. As far as I can see, from this theoretical exercise, two outlined possible scenarios suggest themselves. The current model approaches and considers retinal process in terms of differentiation and thresholds, a process whereby light detection, defined by spikes, its segmentation from noise and initial aspects of scene analysis, are undertaken prior to latter stage assembly type processing in the main brain via the genicular cortical pathway. One critical aspect of the associated retinal processing being how to segment the photon-related spike event from the associated but unwanted noise contained in both the light signal and generated from internal neural processes. However, scientists are increasingly aware that this unproductive noise is counterintuitively not filtered out of the system once segmented. It's retained and appreciated to influence data processing throughout retinal layers in multiple ways. At receptor level, dark light and dark noise are encountered with rod and cone firing at scoptic luminance levels, mediating receptor firing. Largely due to the difficulty in making measurements, debate is still playing out between whether or not its source derives entirely from internal retinal processes or a mixture, including the external incoming signal. To this we have to add the unresolved functionality of gap junctions between receptors, and horizontal cells linking receptors and receptor types located in the outer plexiform layer, together with the amacrine cells performing similar functions in the inner plexiform layer. These cells develop extensive lateral connections and feedback loops that further mediate output to higher layers. It is generally understood that parallel processing system is in play from the receptors themselves, with the cones not simply performing to a binary mode of operation from their two types of synapse with bipolar cells. Large-scale correlated firing of cell groups is also observed, and this is associated with phase-like information being streamed from retinal ganglion cells to main brain areas. With respect to the central hypothesis, it would appear that we have to challenge every aspect of what's thought to be occurring within the membrane, even in relation to receptor function. For example, it's generally accepted that rods are saturated in daylight photopic levels, rendering them inoperable or incapable of contribution to visual awareness. Rods are thought to be productive only at scoptic, dark, or working alongside cones at mesopic, semi-dark levels of luminance. The two scenarios mentioned earlier for retinal processing in support of decoherence and vision space center around receptor function. The first option accepts the duplicity between rods and cones relating to their performance at different luminance levels, with the second being much more radical than the first, namely that rods are actually optimized in photopic conditions. What we have thought of as saturation in terms of a cell's ability to differentiate individual photon detection events being not representative of the principal function, that being noise transmission. Photopic level rod output requires synthesis amongst groups if not entire networks, of receptive fields for subsequent synthesis by amacrine and ganglion cells. We also have to take very seriously the baseline assumption under which current experimentation is designed. Experimentation is organized and calibrated to observe processes in support of differentiation and not synthesis. I've yet to find experimentation that seeks to present the retina with a natural stimulus, a real setting under photopic daylight conditions. It is, after all, in response to these conditions that the eye evolved, enabling us as a sentient being to engage and operate with the environment. If, as vision space theory suggests, we develop spatial awareness direct from the light array in photopic conditions, stimuli that don't supply that specific data formation to the retina will, as an atypical input, alter receptor firing to suit, and so limit vital aspects of signal segmentation. Results taken from experimentation based on the current approach are likely to be incomplete and/or difficult to interpret. So, the first possible scenario. We accept that rods are indeed saturated at photopic lighting levels and that saturation ensures that they play no effective role. However, we accept that decoherence is taking place at the retina and that the phase data potential is embedded within noise. We acknowledge that both rods and cones mediate their firing, indicating the presence of what's termed dark light or dark noise. Given that decoherence would take place within the receptor, we would expect that a debate would result with respect to the origins of that mediating noise. That a second data potential, providing us with a specialist form of spatial awareness, is derived directly from the light array, i.e. is not reliant on conceptualization of the scene from other cues, e.g. occlusion. While we are able to detect the said functional operation involving dark light at scoptic levels as we are dealing with individual light quanta, we would presume that this process is also operational in cones at photopic and mesopic levels of illumination, but impossible to individualize or isolate. The segmentation of the noise-reliant dataset from the photon event dataset would occur via the dual on and off structure of cone synapses, with bipolar cells working in conjunction with the feedback capability provided by the first stage circuitry of horizontal cells in the outer plexiform layer. This first scenario would require that under photopic conditions, we are reliant on the cones in peripheral vision to provide the sparse spatial awareness function apparent in peripheral vision. While this scenario has to be considered as a potential hypothesis, I have issues with it. We are left with either a potentially significant numbers problem with respect to noise-sensitive receptor cell population in peripheral areas, or a very significant total oversight with respect to rod function. If vision space theory is correct, we must assume to have simply missed an entire data structure vital to visual awareness, so I'm drawn to seriously consider this more extreme second scenario. When I structure this argument, it actually makes better sense of retinal circuitry, but also total sense in terms of the experiential reality of phenomenal field. As a non-expert, I consider there to be an intuitive fit. While we have observed that noise processing within the retina is making a contribution to visual awareness, we are struggling to understand what this contribution looks like, how it manifests. I understand from experts that the same is true of the role that decoherence plays in visual awareness. I would suggest that these two factors are directly linked, and that we have failed to make the connection as there's a fundamental misunderstanding enshrined in scientific approach, together with an oversight with respect to vital cross-referencing with intuitive records made of phenomenal field, articulated by visual artists. These factors may have blinded us to a fundamental truth, running right through our understanding with respect to the nature of reality and our place within it. We are partners in the development of visual awareness. The noise from the light array is used, and there's actually a case for suggesting that biological noise may also play a part in the formation of our perceptual structure, being the mechanism through which the visual signals are realized. Vision is a relationship we form with the real. Are we currently living in a hall of mirrors, a situation that persists due to the dominance of the remote observer position adopted by science? Given this set of criteria, we should not be expecting to observe rods detecting from inappropriate stimuli presented to it at photopic levels elements requiring processes involving differentiation. Differentiated responses with respect to exposure to flat, coloured or tonal stimuli would not be tasks rods are designed to perform at photopic luminance levels. They'd be required to passively absorb noise, all of it, without differentiation as phase receptors. If this were indeed the case, then circumstances aligning with saturation would represent the optimization of function and not its cessation or suspension. As mentioned earlier, from analysis at scoptic lighting levels, we understand that cone function also has a component mediating dark noise, and from this we can deduce that they support rod noise development. With their smaller receptive fields, cones would contribute a capability for fine grain synthesis in the foveal area, and to a lesser degree provide some sparse capability for grain resolution in peripheral vision. Rod noise and its associated function at photopic levels would start to fail when illumination levels are unable to deliver a robust enough data potential for streaming. When this condition is reached, we would assume that a reflex kicks in, initiating the well-understood light adaptation period. We would suggest that this transformation process affects the function of the receptors, where they start to align their innate functional sensitivity to differentiation tasks, mimicking aspects of cone function that struggle at these lower illuminance levels. This suggests that spatial vision at scoptic levels is impaired by a loss of the implicit spatial function, and not just by an incremental loss of acuity and colour vision. This again squares with experience, as we regularly knock into objects or misjudge distances, even though we can make objects out within the scene. It also explains the transition in our experiential objective assessments of the moon, through different luminance thresholds. At scoptic levels, the moon looks like a flat shining dish, clear, but entirely lacking a sense of its spherical form. In daylight, it's clearly an object with volume in space. At dawn and dusk, it's possible to make out two distinct monocular images of the object, as the brain struggles to modulate the available datasets to generate a meaningful spatial impression. Towards night, and the conditions of real darkness, one dataset fails entirely. In daylight, the brain becomes increasingly able to resolve the object via constructive modulation, and the double impression sets into an appreciation of its spherical form. This set of conditions is especially apparent with the moon in crescent phases. So the question is, do we correctly understand what's going on with our retinal receptors? Do we understand what's actually involved in an act of observation?